Biopolymers are polymers produced by living organisms. In other words, they are polymeric biomolecules. Since they are polymers, biopolymers contain monomeric units that are covalently bonded to form larger structures. There are three main classes of biopolymers, classified according to the monomeric units used and the structure of the biopolymer formed. Polynucleotides, which are long polymers composed of 13 or more nucleotide monomers. Polypeptides, which are short polymers of amino acids, and polysaccharides, which are often linear bonded polymeric carbohydrate structures. Cellulose is the most common organic compound and biopolymer on Earth. About 33% of all plant matter is cellulose. The cellulose content of cotton is 90%, for wood is 50%. Biopolymers versus synthetic polymers. A major defining difference between biopolymers and other polymers can be found in the structures. All polymers are made of repetitive units called monomers. Biopolymers often have a well-defined structure, though this is not a defining characteristic. The exact chemical composition and the sequence in which these units are arranged is called the primary structure in the case of proteins. Many biopolymers spontaneously fold into characteristic compact shapes, which determine the biological functions and depend in a complicated way on the primary structures. Structural biology is the study of the structural properties of the biopolymers. In contrast, most synthetic polymers have much simpler and more random structures. This fact leads to a molecular mass distribution that is missing in biopolymers. In fact, as the synthesis is controlled by a template-directed process in most in vivo systems, all biopolymers of a type are all alike. They all contain the similar sequences and numbers of monomers and thus all have the same mass. This phenomenon is called monodispersity in contrast to the polydispersity encountered in synthetic polymers. As a result, biopolymers have a polydispersity index of 1. Conventions and nomenclature. Polypeptides. The convention for a polypeptide is to list its constituent amino acid residues as they occur from the amino terminus to the carboxylic acid terminus. The amino acid residues are always joined by peptide bonds. Protein, though used colloquially to refer to any polypeptide, refers to larger fully functional forms and can consist of several polypeptide chains as well as single chains. Proteins can also be modified to include non-peptide components, such as saccharide chains and lipids. Nucleic acids. The convention for a nucleic acid sequence is to list the nucleotides as they occur from the 5' end to the 3' end of the polymer chain, where 5' and 3' refer to the numbering of carbons around the ribose ring which participate in forming the phosphate diester linkages of the chain. Such a sequence is called the primary structure of the biopolymer. Sugars. Sugar-based biopolymers are often difficult with regards to convention. Sugar polymers can be linear or branched and are typically joined with glycosidic bonds. The exact placement of the linkage can vary, and the orientation of the linking functional groups is also important, resulting in alpha and beta glycosidic bonds with numbering definitive of the linking carbon's location in the ring. In addition, many saccharide units can undergo various chemical modifications, such as amination, and can even form parts of other molecules, such as glycoproteins. Structural characterization. There are a number of biophysical techniques for determining sequence information. Protein sequence can be determined by Edman degradation, in which the N-terminal residues are hydrolyzed from the chain one at a time, derivatized, and then identified. Mass spectrometer techniques can also be used. Nucleic acid sequence can be determined using gel electrophoresis and capillary electrophoresis. Lastly, mechanical properties of these biopolymers can often be measured using optical tweezers or atomic force microscopy. Dual polarization interferometry can be used to measure the conformational changes of self-assembly of these materials when stimulated by pH, temperature, ionic strength or other binding partners. Biopolymers as materials. Some biopolymers such as naturally occurring xenon and poly-3-hydroxybutyrate can be used as plastics, replacing the need for polystyrene or polyethylene-based plastics. Some plastics are now referred to as being degradable, oxidegradable, or UV degradable. This means that they break down when exposed to light or air, but these plastics are still primarily oil-based and are not currently certified as biodegradable under the European Union Directive on Packaging and Packaging Waste. Biopolymers will break down, and some are suitable for domestic composting. Biopolymers are produced from biomass for use in the packaging industry. Biomass comes from crops such as sugar beet, potatoes or wheat. When used to produce biopolymers, these are classified as non-food crops. These can be converted in the following pathways: sugar beet and GT, glyconic acid and GT, polyglyconic acid, starch and GT, lactic acid and GT, polylactic acid, biomass and GT, bioethanol and GT, ethene and GT, polyethylene. Many types of packaging can be made from biopolymers: food trays, blown starch pellets for shipping fragile goods, thin films for wrapping. Environmental impacts. Biopolymers can be sustainable, carbon neutral and are always renewable, because they are made from plant materials which can be grown indefinitely. These plant materials come from agricultural non-food crops. Therefore, the use of biopolymers would create a sustainable industry. In contrast, the feedstocks for polymers derived from petrochemicals will eventually deplete. In addition, biopolymers have the potential to cut carbon emissions and reduce CO2 quantities in the atmosphere. This is because the CO2 released when they degrade can be reabsorbed by crops grown to replace them. This makes them close to carbon neutral. Biopolymers are biodegradable, and some are also compostable. Some biopolymers are biodegradable. They are broken down into CO2 and water by microorganisms. Some of these biodegradable biopolymers are compostable. They can be put into an industrial composting process and will break down by 90% within 6 months. Biopolymers that do this can be marked with a compostable symbol under European Standard N 13432. Packaging marked with this symbol can be put into industrial composting processes and will break down within 6 months or less. An example of a compostable polymer is PLA film under 20 micrometers thick. Films which are thicker than that do not qualify as compostable, even though they are biodegradable. In Europe, there is a home composting standard and associated logo that enables consumers to identify and dispose of packaging in the compost heap.